Good afternoon. Welcome to this Spruce Budworm Public Information Session. My name is Logan Johnson, and I'm moderating today's event on behalf of Keeping Maine's Forests. And this event is hosted in collaboration with the Spruce Budworm Task Force and the Cooperative Forestry Research Unit. I'd also like to offer a special thanks to Regina Smith, who's done a lot of the legwork to make today possible. A couple logistics before we get going. All question and answers will be held until the end of the meeting, including on Zoom. So you're welcome to post your questions in on the Zoom Q&A, but they will not be answered right away, as others in the room may have similar questions to you. When we do enter the Q&A, we'll start with the questions that we have collected via Zoom, and then we'll have a microphone working around the Orno room, and then if there's time, we'll follow up with questions up in the Fort Kent room. If you are staying after for some of the tours, we'll have follow-up logistics with you after the Zoom webinar ends. A little bit about the agenda today. We'll start with opening remarks from Allison Kanoti, the state entomologist at the Maine Forest Service, followed by an introduction to Maine spruce budworm history and the current challenges ahead. We'll then go into the budworm monitoring and biology, and the efficacy of early intervention strategies for suppressing spruce budworm outbreaks in New Brunswick. We'll follow that up with a presentation from the Board of Pesticide Control, and then the Maine Budworm Response Coalition, or MBRC, will share their strategies this spring and summer. So without any further ado, I'd like to welcome Allison Kanoti, the Director of Forest Health and Monitoring at the Maine Forest Service and the State Entomologist. Allison first worked for the Maine Forest Service measuring plots in the Forest Inventory Program in Down East Maine. After completing a Master's of Science focused on balsam woolly adelgid, she became a forest entomologist with the Maine Forest Service. Allison's current role encompasses planning, administration, and policy development related to forest health and National Inventory Program.
Good afternoon. Thank you, Logan, for the introduction, and to Keeping Maine's Forests for hosting this event, and also to the planners and contributors for this event, and for the audience for caring and showing up. I want to start with just a little bit of personal context for my remarks today. And there we go, maybe. So I began my path towards a career in entomology growing up on a homestead in northern New England. The journey towards entomology was supported by a rural upbringing, an innate curiosity, and also shyness. I found comfort, entertainment, and companionship from exploring the world around me and learning the names of the others inhabiting it. By the time I'd started my formal schooling in natural resources, when she was just 6 years younger than I am now, my mother's career was just taking off, and she had to suddenly leave it because of an exposure to toxic chemicals in her workplace. To this day, her ability to function is compromised by things like new construction, perfume, air pollution, and other everyday chemical exposures. Moving on to the topic of the day, although it shaped the forests that we work in today, spruce budworm outbreaks have been the stuff of stories and not direct memories for many of us working to respond to the building populations today. Some would say it's the stuff of nightmares. As I was wandering around the fields and forests of my childhood home, examining the life in the cow patties, the brooks, and the forests, a peak of 3.5 million acres of forest a year would be treated in Maine with a mix of carbamates and organophosphates to keep the trees alive for harvest. Sorry. As a response to budworm began to peter out, the first experimental treatments using BTK covered a small percentage of the annual treated area. That's where the nightmare of budworm management starts to become something that a kid who found enjoyment in the insects inhabiting her world and whose mother has been hurt by chemical exposures can get behind. Thanks to the hard work of those who came before us and who recognize the shortcomings of the approach of the day, we are not doomed to repeat these battles. 10 years ago, I traveled to the North Shore of Quebec for my first sight of live spruce budworms and their damage to the forest. The trip was an opportunity to receive a fire hose of information from those tracking and responding to the outbreak in Quebec. It also left with me visions of the consequences of unmanaged budworm populations. Around the same time, the science supporting an early intervention strategy had left the theoretical realm and was being applied in New Brunswick's forests. The budworm response of my parents' generation does not need to be the response of today. A successful early intervention strategy can keep forests healthy, supporting not only the economies that rely on them, including forestry, tourism, hunting and fishing, outdoor recreation, and others, but also maintain wildlife habitat, protection of water quality, and other benefits that our forests provide. Spruce budworm outbreak control under this approach relies on healthy insect communities. For the strategy to be successful, the insecticides used in early intervention must not have long-term impacts on the insect biota of the forests. They must be targeted and break down quickly. The hosts of today's forum, Keeping Maine's Forests, have in their name one of the reasons an early intervention strategy to manage spruce budworm makes sense in Maine today. Maine is blessed with more than 89% forest cover that quietly supports our way of life and provides a backdrop to so much of what makes Maine special. Our forests are predominantly privately owned, but they provide immense public benefits that many take for granted. Every year, Maine forest land faces more pressures for conversion to other uses. In the last reported inventory cycle, the area in forest land decreased by around 0.15% on average per year. Keeping Maine's forests as forests is the key to our future success in Maine. The approach you will learn about today as this program unfolds will help support that goal. Thank you.
Thank you, Allison, for grounding us in today's topic. Our next speaker is Mike Parisio, Forest Entomologist for the Maine Forest Service, who will provide an introduction to Maine spruce budworm history and current challenges ahead. Mike is a forest entomologist with the Maine Forest Service Division of Forest Health and Monitoring. He has worked on spruce budworm in Maine since he started with Maine Forest Service in 2019 and is responsible for organizing Maine's cooperative spruce budworm pheromone trap monitoring program. He also serves as one of FHM's aerial surveyors for spruce budworm damage and compiles the Maine Forest Service annual spruce budworm report, which details Maine's cumulative annual monitoring efforts and tracks local spruce budworm population trends.
Logan. All right, so this is built into one. All right, thanks everybody. Good afternoon as well. So in keeping with some of Allison's points she brought up, I'm going to go through a little bit of some of the funner sides of Maine's spruce budworm history. So I decided to dig through some of our archives for some of the material for this presentation. And, just flipping through a lot of these, just starting to read. So I figured I'd start with an excerpt of a publication, an educational style pamphlet written in the 1970s. And if you, oops, my highlighter disappeared. But anyways, if you go through that, that's kind of a pretty typical description of, what happened in, prior to the 70s, what happened in the 70s, and what we're gonna, potentially be talking about with the current outbreak there. So one of the themes of the content I'll present early on here is that spruce budworm, is not a new thing to Maine. It's part of our history. But as you'll learn from some of the other speakers, the way we think about spruce budworm management, some of the actions we'll take, those approaches are now differently, and they make a lot more sense than some of the things that have happened in the past. But so I was originally going to just do a 50-year look back at 1975, during the maybe 1975, 76 being kind of the peak of the last major budworm outbreak. But then I realized, it's pretty easy to take it further back. So we'll go back 100 years. So as you saw on that first slide, there was a spruce budworm outbreak in Maine and surrounding regions in the 1910s, and some of that, aftermath was carried forward into the 1920s. So you can see in that photo on the left, that's from 1925, I believe itself. So some of the aftermath of all those forests. And again, you can see in the map on the right just the sheer geographic extent of spruce budworms. So one of the things about the 1920s and dealing with the aftermath of that forest. So even though the concept of aerial spraying did emerge as far back as the 1920s, it was in its infancy, used for agriculture for the first time in 1922, I believe. So the concept of spraying for forestry management just wasn't on the table. So a lot of the responses to what was happening in that outbreak would have been silvicultural on the ground management of trees and stuff. All right, so let's jump forward to the next recent spruce budworm outbreak in Maine. So that's looking 75 years ago to the 1950s. So again, same story as you saw in that first slide. Maine does have a history of these returned outbreaks, but we have some differences here in the 1950s versus the outbreak in the 1920s. So that map in the middle there is again just the extent of the outbreak, but we have some new tools that may not be very well understood at this point in time, but they are new and available, and people are going to start using them. So I just want to point out, 1954, you can see in that excerpt there, that was the first aerial spraying program for spruce budworm in Maine to occur. Relatively small, 21,000 acres, treated with 6 planes. But as you'll see here, and I won't get into, but you're all probably aware, one of the products of choice in the 1950s was the dreaded DDT. So I won't again dive into that, but again, that was what was available. That was thought to be something effective and of use, and so that was what was widely used at the time. So let's go 50 years ago into the 1970s. So again, this was an even larger outbreak. I just put some maps in here again to show, we're talking about the spruce budworm situation here in Maine. However, this is always a regional event. That's the nature of these spruce budworm outbreaks. So again, spanning from Ontario to Newfoundland, very similar to, some of the damage extent in Canadian areas we're looking at today. But we're going to talk about, a proposed spray program in the order of about 240,000 acres. You can see there, back in 1976, there were 4 million acres on the table for potential spraying at the time. And again, you can see my slides completely. But, anyways, I'm cut off here on my screen. So some statistics about the last outbreak of the 1970s and stuff. So as far as timeline, again, I took a closer look at 1975, 76, but the whole outbreak, spanned this period of decades, pretty much starting in 1967 with the last traces disappearing in 1993. Again, regional outbreaks, so 136 million acres across Canada and Maine affected. Extremely high mortality rates during this time. So you'll hear from Angela probably, balsam fir is the preferred host, a little bit more susceptible to budworm defoliation and damage. So depending on area, mortality rates as high as 97%, but again, averaging those very high percentages of mortality. Spruce fared a little bit better, but still 30 to 66%, very high mortality rates. Again, one of the the heartburns associated with the budworm outbreak of the 70s is the economic impacts that it had at the time that are lasting to this day even. So an estimated 20 to 25 million cords of spruce fir lost, obviously translating to hundreds of millions of revenue lost to the forest industry. Again, just to harp on the fact, that yes, we're talking about Maine today, but these are always these regional outbreaks. So there's a timeline of, things happening in Canada with Maine included. That red box there just is that snapshot of 1975. So kind of the peak of the last outbreak period. Again, all my notes are missing from this, but I did, go through it, draw little things all over this. So Hard to see now, I did have Maine traced on there, but this is actually a geographic map of Canada and Maine. So if you look at that 26 in bold right there, you know, that's the number of times defoliated between 1954 and 1980. So that's Northern Maine. So even though these outbreaks have been huge and regional in nature, you know, Maine has kind of bore a bigger brunt sometimes, especially during that last outbreak. So obviously during the 1970s, there was also a lot of spraying going on, as you saw from that, you know, 1976 proposed area there. But the strategy of how those sprays were applied was much different than some of the strategies we're going to talk about today. So I just want to bring up this concept of foliage protection, which was the norm at the time. So you can see there, again, Quebec, New Brunswick dealing with this outbreak simultaneously as us. That, you know, excerpt from Quebec on the left, you know, the, you know, amount of acres needing management and spraying just far exceeded anybody's capabilities. So the strategy kind of became to, you know, look to where the damage was worse at that very late point in the damage to the tree, just try to spray it, keep it alive, again, with that end goal of, you know, preserving it as a forest product to be harvested, you know, salvaged and preserve it. So yeah, I won't read through that whole excerpt from New Brunswick, but same thing, just kind of talking about that concept of, you know, keeping a crop alive from year to year for harvest. Again, there will be a whole presentation on the chemical options then versus now. One of the things that I started to notice when I was just looking through the 1970s literature, obviously DDT was used in the 50s. That had been banned by the time the 1970s outbreak was happening. So there was a whole suite of chemicals being applied during the 1970s. And one of the things that kind of stuck out is a lot of these things were just coming on the market and kind of being field tested live. So a lot of them came and went. But you can see there on the right, BT, which we'll talk about again today, which is kind of in its early stages. It had been around for a while, but as far as being used for forestry applications, again, you can see 1978 marked the first time that BT was actually used for forestry management for spruce budworm in Maine. So. And then we'll move on to just a little bit of what we're facing today. So probably everybody has seen that picture on the left in some publication. I actually found the caption that goes along with it. So finally it's west of Chesuncook Lake, but yeah, that's a pretty classic landscape from back in the day, and that still appears in a lot of publications. But this is the situation we're confronted with, you know, today in 2024. You know, these are pictures from aerial survey flown on the Quebec border just this past year. So again, pretty similar. Arrived very fast, but that's what we're getting towards managing. So. And then I just want to talk about some of the monitoring tools that we have today at our disposal that weren't available back in the day. So we'll talk about the fact that, you know, spruce budworm, the location of where it's happening is always tied to the region, and it's not just a matter of it spreading locally. Spruce budworm is transported by huge regional flights, migrations and stuff. So a lot of what we're dealing with today in 2024, 2025 is the result of some very large inflights of moths from outbreak areas in Quebec in really 2019 to start this. But we now have some pretty sophisticated tools to know what these moth flights look like. We can model them using climate, atmospheric conditions, things like that. So we know we are in fact connected to what's happening in Quebec and beyond now. So yeah, as far as Ontario as well. I'll just mention our pheromone trap network. So again, this is a cooperative program between Maine Forest Service and, you know, many other, you know, industry cooperators and public landholders. So I just want to kind of focus in on that area in far northwestern Maine, and we'll just go through the motions to get to that. So here's what the results look for all of northern New England. As you can see, these pheromone traps attract the male moths, and you can already see there's a couple larger points in that far northwestern corner of Maine. So that's the first piece of evidence we had that these populations were expanding, particularly in this area. You can see the results of those pheromone traps again on that green map of Maine on the right, so those higher numbers there. And then we still do aerial survey with airplanes and helicopters, but a lot of other groups have moved to satellite imagery. So in Quebec, they're modeled in those 3 different colors. Those are results of, you know, the foliation surveys from their analysis of satellite imagery. So as you can see, that orange zone right there near those, you know, high pheromone trap captures has hit the Maine border and obviously has not stopped. So building on that, again, you know, we're still looking for damage from the air for, you know, using airplanes and human observers and stuff. So going to that area, you can see there, the pink polygons on the map on the left and the yellow on the right, those are the same areas, but that's, you know, physical, visible defoliation like that picture from out the airplane window. So for the first time in many years, we had a significant amount of visible damage, you know, to the naked eye amounting to about 3,500 acres. So yep. And then we also follow up with a lot of ground defoliation survey, again, using a method that is tried and true that dates back to the 50s, I believe, the FETIS method. So again, just looking at that particular area of interest, you can see those numbers really expanding on the Quebec border there. And just for some context, the 2 years previous, you know, those defoliation rates were, you know, below 10% both years. And so you can see we have areas that are as high as 70% defoliated. So obviously something going on right there. And I'll talk about, or Angela will talk about how we know even more about this stuff. So that's what I have just to set the stage, present you all with the idea that, you know, budworm has happened many times in Maine, it's happening again, but our approaches will be different. So what you'll hear next from Angela, you'll learn about, you know, our most sophisticated and best monitoring tool and how we've started to incorporate that into our management approaches. We heard from some friends in Canada that have been dealing with budworm longer in the modern age than we have some new ideas and approaches. So we'll learn about some different management techniques. Again, there's going to be a lot of talk about the chemicals being used, and we can talk about advancements in those and the differences between what was used back then and what we're going to use right now. And then, yeah, you'll hear a little bit about, you know, how the team has been organized to kind of take this on together as a cooperative effort. So that's all for me. Thank you.
Thank you, Mike, for providing some background and context for today's situation. Our next speaker is Dr. Angela Mech, the Assistant Professor of Forest Entomology in the School of Biology and Ecology at the University of Maine. She is also the director of the Spruce Budworm Processing Lab, which opened at UMaine in 2021. The lab works with landowners and stakeholders to monitor spruce budworm populations in New England, dominantly Maine.
Thank you so much for inviting me, and thank you all for coming out today to learn a little bit more. So you just had your history lesson, and now we're going to move on to biology class. And yes, there might be a quiz. So spruce budworm, it is known as the most severe defoliating pest of spruce and fir in North America. I didn't give it this title, somebody else did. And what's interesting is normally when we think of destructive insects, we think of invasives. But this one is actually native. And so it is from North America, and its range, basically you can find it wherever we have balsam fir and spruce. The map on the right is spruce fir forest concentrations within the US, and you can see that Maine is a little bit darker than the rest. And then balsam fir range, you can see, is almost transcontinental, and so you can find eastern spruce budworm going all the way across. Like Mike mentioned, it does experience cyclical outbreaks every 30 to 40 years, and this has been happening for thousands of years. This is actually part of their normal behavior. And from an evolutionary standpoint, it's actually pretty cool. They basically wait their turn and then explode so much so that nothing can stop it. And then they go back to their quiet little normal lives for 29 more years. So it's similar to the cicadas, right? Periodical cicadas, they take 13 years, 17 years, and then they're just everywhere. And so spruce budworm is similar to that. Luckily for us, it only has one generation a year, so we're just dealing with feeding occurring at this one main time rather than having multiple generations overlapping. At first, we're gonna have the egg masses. So these are gonna be laid, the females can have up to 200 eggs, but she's not gonna lay them all together in one massive clump. She's gonna do 20 here, 20 there. They're gonna be laid on individual needles on fir or spruce trees. Then they're going to hatch a couple weeks later. And these little baby caterpillars we refer to as first instars. And at this point, they're basically just moving around trying to find their future home. So some of them might stay on the same tree where they hatched. And some of them do this cool, like we call it ballooning, where they'll take a silk thread, they'll come down, and then they'll just get blown in the wind and just see where they land. And so actually during this first instar stage, we can have some pretty high mortality in spruce budworm if they don't end up where they want to. And the key thing here is that they don't actually feed when they first hatch. And so once they find that suitable spot that they want to take a nap in for the winter, they molt, like all insects molt during these different stages of its life cycle. And so they turn into these second instar larvae, or L2s as we call them. And so once they enter the L2, they're gonna make a little silk home and some nook and cranny on the branch, and they're gonna go through hibernation for 7 to 8 months. So basically from late August through till about now. Once April comes around, they come out of their winter hibernation, they're gonna start feeding, but we don't quite yet have the new growth, so they're gonna feed on the buds, they're gonna feed on the pollen cones. And so they're waiting for that new growth to flush out because new growth is nice and soft. And so for their fresh young little mandibles, it's much easier to chew on the softer needles than it is the older, harder needles in the back. And during this time, we refer to them as kind of messy eaters. So as those needles are emerging out, they're gonna chew on a needle a little bit and then move on to another one without finishing the whole needle. So the needles turn brown, they stay in there, it's wrapped in silk with their poop in there too. And so you get these scorched tips on the branches that tell us that, okay, spruce budworm is in there feeding. They're gonna pupate then in early July, so most of our massive feeding is gonna be occurring in June. And then we have adults a few weeks later, mid-July and August. And the adults are fairly small. So you can see that's somebody's finger, moth is on the finger. So they're pretty small. They're gonna have those 2 lines that are highlighted. These 2 bold lines on their wings is how you know that you're looking at a spruce budworm. Okay, like I mentioned, these guys outbreak, and they're not the only ones. So what are these stages of an outbreak? So first we have this non-outbreak phase. And truthfully, when they are not outbreaking, we can't find them. They're very, very low densities. They're kept in check. And yeah, we don't see them. And then there's this building phase. And the building phase normally goes unnoticed because we haven't yet seen this extreme damage on the landscape. Then once we hit the outbreak phase, that's when we see the defoliation on the landscape, we see tree mortality, we begin to panic. And very often by this point, you're dealing with a much larger issue than when it was just building. You'll have the decline phase. That could be just because they ran out of hosts. Every outbreak does eventually decline. Depends on the species, it can last 3 to 5 years, or in this case, a decade or more. And then in that non-outbreak phase, they're basically kept in check by natural enemies. So these are things in nature that keep spruce budworm down. So birds love their caterpillars. We have parasitoid wasps that will attack them, predators, and then there are even viruses and things that can go through the population. So as Mike mentioned, the outbreaks can last 10 or more years. And so even though they prefer to feed on the new growth, during an outbreak, they kind of have no choice. They have to start feeding on the older needles because the trees are going to stop producing new needles if they're really stressed out and they've been chewed up. And so this picture is really kind of cool because you can see all the different stages. So if you see the gray tree, that's obviously has been killed by spruce budworm. The one kind of northeast of it, That one has been, the inner needles have been chewed up as well as the outer. There's no more new growth. That tree is basically going to be fully dead within another year. To the left of the gray tree, you see the scorched, but if you look closely, the inner needles are still green. And similar to the tree to the northwest of it is also scorched tips, but still green on the inside. Those trees can actually still be saved. It's just the new growth that has been attacked. And so trees do have the ability to regrow and to tolerate some herbivory. It's just when it becomes too much, the older needles start getting chewed up. That's when we begin to see mortality. About 3 to 6 years of that sort of defoliation, we're gonna have mortality on the landscape. And so during the last outbreak, it was up to 97% of the fir and up to 60% of the spruce. So like Mike mentioned, I don't know who named it Spruce Budworm, but it should have been balsam fir budworm or something, because they really do prefer the fir over the spruce. Okay, so during the outbreaks, it's eruptive, it's massive, we have trillions of them. This is actually a quote from the last outbreak. And this is talking about the adult emergence, when we just have moths everywhere. They said, in places they died in masses and could be shoveled like snow. Crushed bodies made road traffic hazardous. Clouds of moths caused traffic to stop. And in July, coastal fishermen reported 20 to 50 dead moths per square foot of ocean miles out from shore. And so the Healthy Forest website here has some really cool videos, and so I just took some images from it to show one of these massive influx of the adults. And so it looks like she's just going to grab like maybe some wood chips at the bottom of that tree, but that is just masses of adults that have basically probably laid their eggs and then just died below, and you could see she just scooped them up, and it was just this trailing dead moths just everywhere. So it can be pretty extreme when we have these events. And also, like what Mike showed, you can pick them up on the radar. So when there are, when the adults all emerge at the same time, they can travel hundreds of miles by going up into the wind currents. And when there's a lot of them, it can actually be picked up on radar. And so this was from June 19th of this past year. And you could see that kind of some of them landed right in the southwestern part of Maine, not where we were wanting to see them go. But at least we weren't New York. A few days later was a bigger emergence. And it was as if they just went right to the Adirondacks. So it'll be interesting to kind of see what happens to the budworm population, not only in Maine, but in other parts of New England and now New York. And so although they can come in on these big immigration events when we have this, so coming in from Canada, there's also kind of the spillover of the hotspots growing and then getting into new areas. And so like Mike mentioned, New Brunswick is doing something really cool. And this is like a prime example of just like, whoa, this is really cool. It's as if Spruce Budworm got to the New Brunswick border and said, nah, never mind. We don't want to go there. Although, why wouldn't you? New Brunswick is beautiful. And so it is because they're implementing a really cool strategy that we're going to learn about next from Sara. But it's kind of comparable to our Chuck E. Cheese fun game from the 80s, where as soon as something kind of looks like it's popping up, you just knock it back down. And so it kind of asks the question of, what if we could actually prevent that outbreak phase of an outbreak? So instead of waiting for that peak moment at the top of that curve and panicking, what if we could actually stop it before it reaches that point? So this is kind of the fundamental question that we're asking. And so in order to do that, you kind of need to find that point, right? We have to be able to monitor to see, is the population doing that? Is it up here? Is it down here? Where is it? And during the last outbreak, they monitored using egg mass surveys. So this is a picture of all of the volunteers. It happened to be a lot of foresters' wives and folks in the community, because egg masses, they're only egg masses for like a couple of weeks. So in just a few weeks, they had to monitor the entire statewide population by looking at needles for tiny egg masses. And I'm sure you can imagine, gee, that must have been a lot of fun. And so it was a very short window. There was chances of human error. If it was a low density population, you know, it's like looking for a needle in a haystack. And so this wasn't necessarily, it was what we had at the time, but we can do better. And so now, at the end of the last outbreak, they came up with this new method to monitor that was way more efficient than doing the egg mass surveys. And this is the L2 sampling. And so this is looking at that second instar caterpillar that is hibernating for that big chunk of time over the winter. And so this is actually a good indicator of the population that is going to be in that stand. Because remember, the eggs, they can hatch and leave, or they can hatch and die. And so it might not actually be a good indicator of what's in that stand, but this is. These are gonna wake up in the spring and start feeding exactly where they are. The other thing is that we can sample now for 7 to 8 months rather than trying to squeeze it all into a few weeks. And this gives stakeholders and landowners opportunity to figure out, you know, what they're gonna do. And then it's relatively cost efficient. We're basically just paying for consumables to do the processing for this. So what is this process? Okay, so it starts, our first step is that we have a whole cohort of volunteers, which are our landowners and stakeholders, that go out to their land and will clip a 30-inch branch from 3 trees in a location, 1 branch per tree. And it's gotta be from the mid-crown. So these folks are out there with what, like 3, 4 extenders on the pole pruner, sometimes 5, to get branches from the middle of each tree. And then they bring them to the lab for processing. And so we take each branch, gets its own little bucket, gets clipped up, and then we soak it in sodium hydroxide for a couple of hours. And this is gonna dissolve the silk and basically release all those larvae. Then we filter out all the plant debris because we don't wanna be having to shuffle through needles and branches. We have another fun stage that I just call chemistry magic. But we separate out the larvae from additional debris. Then we get it on a petri, we put it under a microscope, and we have to identify all these larvae. And so these caterpillars are tiny. And so each table actually has, I put a vial in the middle of each table so that you can see the size, an actual size of the L2 larvae. And so they are extremely small because they just basically hatched before they went into their diapause. And so we have to look at that tiny of a larva to try and figure out what species it is. And this normally gets some chuckles, but in order to identify larvae that small, we have to look at their anal comb. So yes, that is a biological term. There are caterpillars, you know, if you think about what comes out that end, you don't want it to stick to your butt. So they have these little combs that can help flick away their waste. And those combs have different shapes and sizes to them. And so we have to look under a microscope, get the position correct for each caterpillar, and look and see if its comb looks like a crown, which tells us it's spruce budworm, or are the teeth of the comb different sizes, different heights, different widths. Then we might be looking at other caterpillars like we often find fur combworm on the samples as well. So once we do all the IDs, if we end up with an average of 7. So if we have an average of 7 of those L2s on the 30-inch branches, that tells us that that location is going to be dealing with a potential problem in the spring when they all wake up. So our L2 monitoring here in Maine, we opened the lab here in 2021. We were funded by the CFRU as well as the US Forest Service. And we've been monitoring about 300 sites in Maine, and then we do usually another 20 or so in New Hampshire, Vermont, and this year we started New York. And we have a fee for service option. So if landowners, for example, we find a hotspot, it's over 7, they can go back out and do radial sampling to determine, well, how big is that hotspot? In case they want to determine a management plan, they need to know how big it is. So we offer this fee for service option. So cost efficiency, it costs about $35 per site. So for 3 branches to get processed for that. And so this is our trends for the last 5 years. And as you see, kind of low, kind of low, kind of low. And so this is kind of fitting that curve, right? So the curve I showed in the beginning, we're seeing that uptick in the population. And so this is across Maine over this time period, and you can see that the number of sites that have had a hotspot, we only had 2 last year. And this year we found 21. And so the population overall is sort of going up, though it does seem to be concentrated in certain areas. So the L2 data that comes out of the lab here, we share that with my colleague Neil Thompson of the UMaine Fort Kent, and he does his modeling spatial magic. And so there is a statistical model that is used to try to estimate, okay, so if we know this spot is this density and this one is this density, what do we think it is in between? And we're able to come up with this map. And so we make new maps almost every single week during the processing season to kind of figure out the size of these hotspots. And so this year we did a little over 700 sites in Maine, and we found a little over 300,000 acres that are at risk that have these above normal spruce budworm populations. So kind of setting the stage, now that we know that we have this issue, we're going to talk next with our Canadian neighbors to hear how they tackle the above normal spruce budworm population. So thank you.
Thank you, Angela, for taking us back to biology class. Really appreciate it. We're gonna just take a couple seconds to switch over screens as we move to our friends over in Canada for their efficacy on early intervention strategy for suppressing spruce budworm outbreaks. Our next presenter is Sara Edwards, a research scientist at Natural Resources Canada. She has been involved in budworm research since 2019 when she joined the early intervention strategy research team. Her work focuses on evaluating treatment efficacy in early intervention. So, Sara, you can go ahead and share your screen and I will spotlight you for the crowd.
There we go. Sorry, couldn't find the unmute button. Is that showing the slide view, and can you hear me?
I can hear you and see your slide view. I just need to make you viewable from the audiences.
This is perfect. Gives us some time. Everyone can watch this little animation run through with the defoliation across Canada, kind of over time in our history with budworm.
You can go ahead and start presenting whenever you are ready.
Perfect. So hello everyone. As mentioned, I'm going to discuss the early intervention strategy that we've used here in Canada to control budworm, largely in Atlantic Canada. And first, I'd quickly like to acknowledge that I'm part of a large research team here working on budworm, and what I'm presenting today is a huge collaborative research project. This involves many different scientists, technicians, countless summer students, and from across different organizations. And without all of these people, this research would not be possible. So moving into budworm. It's going to come as no surprise to everyone here that forest disturbances like forest fires or pest outbreaks can impact both the ecological and economic functions of a forest. So this includes things like carbon storage, ecosystem services, timber production, recreation, tourism, and the list goes on and on. When disturbances occur, if we choose to intervene, we need to decide what type of strategy we're going to use to mitigate the effects. When it comes to forest disturbances, there are two general strategies we can take. The first control The first is a control strategy. So with a control strategy, we try to suppress the disturbance and contain its spread. So you might want to think of a small wildfire. We might decide to extinguish the flames. This prevents its spread and keeps the damage relatively small and contained. So we manage to suppress and control that fire. The second strategy we might use to deal with a forest disturbance is a protection strategy. So with a protection strategy, what we would do is let a disturbance run its course and protect the most high-valued resources in its path. So if we think again of that kind of wildfire example, a protection strategy may be used if the fire is very large. We might try to protect valuable resources like housing and infrastructure, while the fire is left to run its course elsewhere, as containment and suppression is extremely difficult and firefighting resources are limited. So we adapt a different type of strategy. In forestry, pest management largely favors a protection strategy. So actions are taken to protect high-valued stands, and outbreaks are allowed to peak and decline naturally elsewhere. This has been the main strategy used to manage budworm. So we call this foliage protection strategy, and you've heard this term already before today. Just revisit it for a minute here. So the goal is to treat the high-valued stands of fir and spruce and reduce the defoliation caused by this pest to about 50%. And this is done in an effort to keep the trees alive until harvest to really reduce the economic losses. Budworm populations, defoliation, and tree mortality are high everywhere else, kind of like our raging wildfire example. So this protection strategy is used largely because control efforts, where we tried to suppress budworm populations and contain the outbreaks, they failed in the past when we attempted them before. So although the efforts to achieve population control failed in the past, we thought it was time to revisit this idea of population control. So the last time we attempted to do this kind of population control was in the 1950s. And since then, we certainly have a much better understanding of population dynamics and how outbreaks spread. And since that time, we certainly have seen huge advances in the treatments we use to control budworm and the technology we have to monitor populations. So we've certainly moved past the days where we used to use broad-scale insecticides and hand-drawn maps to manage budworm. And now we're using target-specific bioinsecticides, and we have access to a whole host of monitoring and modeling tools at our disposal. So Quebec is in the middle of a large outbreak. This is causing significant damage across the landscape to fir and spruce trees. So this map is a bit older, it's from 2017, but you can see the extent of the budworm damage was causing at this time. So it's a little bit blurry here, and I apologize if you're colorblind, but this is the light, moderate, and severe defoliation that was evident across the landscape. And based on our knowledge and how budworm spread and these outbreaks move, we were concerned for our forest. This outbreak was, right at our doorstep. So currently, the strategy used to manage budworm in the area where the outbreak is quite large is a protection strategy. So high-valued stands in this area are treated with insecticides to keep the trees alive, largely until they can be harvested, and this is to help reduce the economic losses. In Atlantic Canada, we could see the outbreak at our doorstep, and the decision was made to intervene early and attempt to control populations before the outbreak. And we called this the early intervention strategy. So the early intervention strategy, this is a novel approach to budworm management. This is something new that we were trying. This is area-wide management for population control. So we're trying to manage across the entire landscape by applying targeted treatments. And of course, we monitor for efficacy and non-target impacts. And so how exactly does early intervention strategy work? First, we had to think about how the outbreak would start and spread over our area. So there are a few theories on how outbreaks spread. I'm certainly not going to get into all the details on those. But the most likely theory was this epicenter hypothesis, and that's really what we're working with. So what does that mean and how does that work? So first, moths move from the outbreak area and migrate into our area. They lay their eggs and they bolster the local populations, just bringing them up a little bit. But these populations continue to grow, and as they become larger, eventually they become moth emitters themselves, and they create new hotspots. And this would continue until the moths had moved across the landscape. So what early intervention strives to do is to target these pockets of populations experiencing growth, so these hotspots. And the goal here is to keep them small, inflict a small amount of mortality, enough to knock those populations back down to the natural non-outbreaking levels. And this is where the natural processes can keep them in check and prevent them from outbreaking. So in our very scientific terms, as Angela mentioned before, what we're trying to do is play whack-a-mole across the province. So as these hotspots come up and these local populations are boosted up a little bit, we want to tap them back down and keep them in check. And it's important to mention here that early intervention is not an eradication effort. We're not trying to eradicate budworm. We're just trying to keep those populations at non-outbreaking levels. So what is a hotspot and how do we find them? Hotspots, just as a reminder, are areas where those young budworm reach densities of 7 or more per branch. That's where the research suggests that we will see problems and those populations will continue to rise. So here's a map. So this includes New Brunswick, part of Maine, Nova Scotia, and PEI. So we, of course, sample across the landscape, and this was back in 2016. And this is really just to give you an idea of the sampling intensity and how we are sampling across the landscape to manage across the landscape. So these branches are collected and processed, as Angela described, and any hotspots we found, and I'm talking about New Brunswick here, although we did sample in Maine and Nova Scotia and PEI, but all the hotspots we found up until this point were largely in New Brunswick. So all the hotspots found were treated with insecticides in the following spring. So as we found hotspots come up in New Brunswick, we would treat them to help tap them back down again. So what does that look like? So I'm going to give you an overview of our treatment program in New Brunswick. So from start up until today. So all of our hotspots found in New Brunswick are wrapped in these treatment blocks, which you can see in yellow. I didn't add all the points just to simplify the maps. The points in Quebec are simply to show that we have a lot of pressure from populations just to our north. We call these, I call them hotspots at our border, but densities in some of these spots are over 100 per branch. So these populations are quite high, which puts a lot of pressure on us. In 2014, we had our first hotspots detected, and these were treated in the spring of 2015, which is shown in these treatment area blocks. 2016, the area shifted, relatively small. Like we said, this game of whack-a-mole. So we knocked them down 2015 and we had new ones pop back up. 2017, so we experienced a large migratory event. We had moths move across the province. This led to an increase in the spread and treated area. And this was a bit concerning because, again, early intervention was new. We didn't know how this was going to play out. So 17, we had large treatment blocks. 18, we were still dealing with the aftermath of a migratory event with new hotspots still arising. By 2020, they've reduced back down again, so we're starting to see the effect of this whack-a-mole game. They came up and we tapped them back down, so it looked like what we were doing was paying off. 2021, again, those blocks just shift, and we start seeing quite a bit of reduction in the problematic areas in New Brunswick in 2022. 2023 was extremely small, and 2024. And for this year, this is the draft version of our maps, not the finalized. It looks like our populations have come back up just a little bit, but again, we're just going to continue this cycle of finding them, dealing with them, and keeping them in check. So over time, the total land area treated kind of looks like this. I plotted it here. We start low. We had our migratory event. The area we had to treat increased. Then we dropped back down again. We had a little bit of a second cycle come up. But overall, we kept the problematic areas low. We kept those budworm populations in check, and we prevented spread across the landscape. There are two key questions with early intervention. First, how much mortality are we adding into the system, and can we offset migration? So budworm have a naturally high mortality rate. Most larvae die from predators, parasites, disease, and so on. These can also be affected by things like tree defenses, inclement weather, and so on and so on. In our control sites, budworm mortality as they go from larvae to adult moths is around 80%. So this is just natural mortality. It varies from year to year. Overall, it's quite high. So here I've plotted the average mortality for each year of early intervention. In areas where treatments are applied, mortality rates are generally only about 10 to 15% above what we see in our control sites. So we've added another slice to this mortality pie, and we've reduced the number of moths coming out of this system. So that's exactly what we wanted to see. It's not a lot of mortality, but we don't need a lot. We just put in that extra tiny little bit to knock those populations down and keep them in check. And I'd like to point out that nature takes care of most of this work for us. So we really don't want to disrupt these natural processes and these natural sources of budworm mortality. Our treatments reduce the total number of local moths, but as we mentioned, not all moths are local. The next big question was, can we offset immigration? So these immigration events can be sizable. As pointed out before, so here are some pictures of the mass migration event that we have. The weather radar in the top corner suggests there was a snow event in July. So this was actually a large cloud of moths that moved into our area from Quebec. And if we can't overcome this migration, our treatment effects would really be in vain. So if we look at our treatment blocks, the population densities, so these include the local moths, so local larvae hatched from local moths, and these also count for any migrants in the area. So this is all the larvae combined. In each year, we see that the average budworm densities are well below the threshold we have set. So in our treated areas, these densities would have started above the threshold. After treatment and even after immigration, it looks like those populations are down below that problematic threshold level. So this suggests that we are not being overwhelmed with immigration pressures, and treatments can be used to reset those hotspots to below the treatment level thresholds, which is where we would like to keep them. How does early intervention compare to foliage protection for budworm management? So if we look at the outbreak progression, using two relatively equal-sized land areas from each program, we can kind of look at how these two programs played out. You've seen this plot already. It just shows the area treated per year over time under early intervention. So year 0 represents the year we first saw hotspots detected in the area. With early interventions, we started treatment immediately at year 0. With foliage protection, treatments for foliage protection didn't start until defoliation was observed. So this was 4 years after the first hotspots arrived. Had we not adopted the early intervention strategy, we would likely be in a similar situation. So 10 years into the outbreak, early intervention has overall lowered the treatment area, so the amount of land treated, compared to a foliage protection. So what do the densities look like? Average densities remain low under EIS. You can see them plotted here in the bottom. So these are average densities in EIS, quite low. To our north, average population densities quite high, over 20 per branch, but individual sites could see, well over 100. So this is averaged across the whole landscape, but in individual spots, it can reach incredible densities. And of course, budworm love to eat. So this means large amounts of defoliation and tree mortality. So if we look at the defoliation, across the landscape. Under EIS, I actually had to plot the zeros or else you don't see any lines here. So most of these years we didn't see any defoliation at all. In year 6 and year 7, we did have a little bit of noticeable amounts of moderate defoliation that we could detect, but very small and very limited in the area that was affected. With foliage protection, defoliation across the landscape, I mean, we get considerably high numbers. These are square miles of defoliation. So large amounts of defoliation across the landscape came down, but again, these populations, we had a drop as well, and things are coming back up again now. So had we adopted a foliage protection strategy, northern New Brunswick could have experienced similar densities, so extreme densities of L2 per branch, and that would have led to considerable amounts of defoliation across our landscape. So everyone always asks, why didn't Quebec do EIS? I mean, it looks like it works. Why didn't they do it? I'd just like to point out that this outbreak started well before early intervention was even conceptualized. So they didn't have an early an early intervention strategy to adopt. It didn't exist. The Quebec scientists actually worked with us on early intervention, and they helped to develop this concept. Atlantic Canada was really just lucky. So we saw what was coming our way and were able to intervene. And that's and that's kind of good for us, I guess. So early intervention, is it working? The results are encouraging. We're managing to keep the densities low. A little bit of additive mortality seems to go a long way. We don't need to get 90, 100% mortality of budworm in our treated area. We really just need to add about 10% more, and that's enough to keep those populations in check. Is it sustainable? Really, we just need to see if we can outlast this ongoing outbreak in Quebec, because most of our moths and most of our populations are being bolstered by these immigrants from the high density population. So only time will tell, but we think the results are quite encouraging. And to date, we really have low damage and low tree mortality. And I think that's all I have for you guys today. Normally it'd be questions, but I think we're saving those for the end.
Thank you, Sara. And bear with me as I try to control Zoom screens. Thank you, Sara, for sharing your experiences in New Brunswick. Our next speaker is Doug VanHoewyk, a toxicologist with the Maine Board of Pesticide Control. He assesses the toxicity and risk of pesticides that are registered in the state of Maine. This includes examining and predicting the risk of pesticides to humans, wildlife, and the environment. In this role, he works and communicates with various stakeholders, including farmers and citizens of Maine.
Thanks, Logan. And if I could add to that, working with Mainers is certainly the most enjoyable aspect of my job. Whether it's foresters, farmers, or just the citizen who has a question. So this presentation is going to discuss the two active ingredients that will be applied to contain the spruce budworm outbreak in northern Maine. Toxicologists use the best available scientific data to try to assess and predict risk. And when I talk about risk, here I'm referring to the product of both the hazard as well as the exposure to that hazard. So on the top right, top portion of the slide, I have a whole bunch of pictures of things that might elicit fear, right? Naturally so, right? And so on the very top right is DDT. DDT was a dangerous insecticide applied more than 50 years ago. DDT today is just as toxic as it was 60 years ago. What's changed, however, is our exposure because it's been banned since 1972. And since it's been banned, our exposure decreases. That's going to then, therefore, decrease the risk. Sharks elicit fear, right? This great white shark. If we just swim in the Penobscot River here locally in Orono, the risk, the exposure is negligible, right? But if we go swimming on nearby Acadia, it might increase it to minimal risk. And if we go swimming in Myrtle Beach, South Carolina, now we're greatly increasing our exposure and consequently the risk. Deer ticks, well, in Maine, we know that we're exposed to deer ticks a lot, right? So that increases our exposure, increases the risk. The only one here that I should have maybe elaborated is the cup of coffee, right? I decided to keep this in. In 1991, the scientific evidence suggested that coffee is no longer, quote, probably carcinogenic, right? So prior to 1991, some agencies, the International Agency of Research on Cancer, deemed it probably carcinogenic. That's now changed, right? This reflects the fact that toxicology profiling is an ongoing, active process. What we know today is different than what we knew 50 years ago, 70 years ago. And again, that also applies to DDT, right? When we talk about pesticides, exposure can be via three main routes. It can be dermal, this is contact on skin, it can be inhalation, and there can also be oral exposure. And when we refer to oral exposure here, we're referring to both consuming drinking water as well as a diet. Okay, this slide is going to broadly talk about what makes an effective pesticide. What are chemists trying to do? Well, first, they want to solve the problem, right? And in this case, that problem is the spruce budworm outbreak in northern Maine. So chemists want to design a product, a molecule, that can be applied at low concentrations. It's not going to be efficient to try to apply 100 pounds of a product, you know, per an acre, right? That's going to increase the exposure, and it's just not practical. Similarly, chemists want to make sure that the effective insecticide is applied minimally, right? Once or twice a year is ideal. It's not going to be practical to apply every Tuesday and Wednesday, right? Furthermore, that would just increase the exposure if we have to apply the pesticide twice a week. Importantly, the chemists who design pesticides also are mindful of potential unintended consequences, or in this case, adverse effects. So it's vital that these are minimized. And so when we refer to adverse effects here, we're discussing toxicity to off-target organisms, humans, wildlife. Maine is fortunate to have great wildlife, right? We have bears, we have moose, we have foxes, raccoons. So we want to make sure that wildlife is not threatened. We want to make sure that aquatic organisms are not harmed or other insects. And we also want to make sure that there's low environmental impact, right? So that the pesticide does not volatilize, move into the groundwater, accumulate, or persist in the soil. Okay, So when the chemists started devising a pesticide that would be active against spruce budworm, they wanted to leverage the biological understanding of that pest, right? So in order to make a pesticide that's specific to that target, we want to know what's unique. What is something that we can hone in on? What is a process in that organism that is not shared with other organisms? And one of the things that we know about spruce budworm is that it goes through different metamorphoses. At the caterpillar stage, additionally, it also is going to molt. And this molting is triggered by a hormone. And if you think about hormones, they have to be tightly regulated, right? So it has to be very precise. The amount can't be too high, it can't be too low. It has to be delivered at a perfect time. It can't be delivered prior to molting, or I should say, it can't be delivered well before or well after the intended application or the amount of this hormone, right? So it has to be very tightly controlled and regulated. So shown here on the right-hand portion of the slide is a molecule called ecdysone 20. This is the hormone that's endogenous or natural to spruce budworm, right? I'm going to go ahead and represent this hormone as a red triangle. And again, this hormone is only going to be found in caterpillars and moths. And when this hormone binds to the receptor in these organisms, and again, this receptor is only found in the caterpillars of moths and butterflies. When it binds to this receptor, the receptor here is depicted in blue, it's going to elicit a cellular response. And in this case, the cellular response is molting. And the chemists took advantage of this knowledge, right? They know the structure of this hormone called ecdysone 20. And they then made another chemical that is going to mimic the natural hormone. This chemical is called Tebufenozide, the active ingredient. So Tebufenozide is structurally and chemically very similar to the hormone. And therefore, Tebufenozide, which I've now depicted in green, a green triangle, and again, I'm using a triangle to model the fact that they have very similar shapes, both the natural hormone and the insecticide. Both of these can bind to the receptor, but Tebufenozide has higher affinity or attraction for that receptor. So it's about 70 times more potent than the natural hormone, and it's irreversible. So once Tebufenozide binds to the receptor, it's going to stay in place. And I suppose it's analogous to a lock and a key, right? So the receptor has a specific shape. Tebufenozide has a specific shape that's only found in these caterpillars or caterpillars of moths and butterflies. And that's going to then result in premature molding. It's going to dysregulate the natural development and the physiology of the spruce budworm. So it's going to perturb its life cycle. And this premature molting is going to cause lethality. And you might be asking, well, there are other organisms that mold, right? What about crabs, crayfish, right? Don't they mold? They do. However, but during the course of evolution, crabs, other aquatic organisms that mold, they have evolved different types of receptors, different types of hormones, right? So they don't have the same hormone that caterpillars do that promotes molting. So that's how we ensure some specificity. All right. This slide looks at the risk assessment of Tebufenozide for humans and mammals. When we look at toxicity, this has been determined in a laboratory setting, and scientists use model organisms to determine toxicity, right? So we can't use humans, That's unethical, right? So, so the data that I'm showing here is taken from the EPA. The model organism that's used is rats. And I'm introducing a concept here called the LD50. This LD50 stands for the lethal dose for 50% of the population. So if you look at the table on your right, cyanide's at top. The LD50 is 3.6. So 3.6 milligrams per kilogram would indicate that at this concentration, 50% of the rats would die. And so this is followed by DDT, which is 87, aspirin, table salt, these are 2 household items, and then Tebufenozide. So the LD, and so we can compare these values to the table on the right. This table is taken from the EPA. So the EPA has devised categories, right? So we can match the LD50 with its perceived toxicity. So cyanide is highly toxic. That makes sense, right? That's everything that we know about cyanide. DDT and aspirin, this is going to be moderately toxic. Right there. Salt, slightly toxic. Tebufenozide, practically non-toxic. And not is it only practically non-toxic to rats, but also to humans, birds, mammals, earthworms, and reptiles. For other organisms, risk assessments have been made. So of concern is honeybees, right? So these are pollinators. They help maintain our food supply. The LD50 for honeybees is 234 micrograms of Tebufenozide per bee. So again, we can refer to these published tables by the EPA to help decipher what that means. So for honeybees, 234 is going to be certainly greater than 11, so it's practically non-toxic to honeybees. So in contrast, however, these LD50 assays demonstrate that Tebufenozide is slightly to moderately toxic to some aquatic life, trout, water fleas, and lobster. However, the risk to these aquatic organisms is minimal. This is for 2 reasons. One, the applied concentration is going to be much lower than the LD50 concentration that was determined in these laboratory settings. Secondly, the exposure should be minimal, right? This is certainly true for lobsters. Lobsters are what, 200, 300 miles away from where these active ingredients will be applied. There's also going to be a 100-foot setback from streams and water surfaces. So that's going to help, again, further reduce the exposure that trout, water fleas, water midges might be exposed to. Lastly, we know that spruce budworm caterpillars are not the only caterpillars that inhabit Maine forests. So it's quite likely that other non-target caterpillars could potentially be affected by Tebufenozide if they consume it. The chemical properties of Tebufenozide suggest that there's going to be relatively low risk to the environment. So the Tebufenozide will not volatilize. What this means is that if it's applied to fir and spruce needles, it's not going to move from these needles into the atmosphere and then be drifted off elsewhere. That's a good thing. Tebufenozide does not dissolve well in water. What this means is that it lacks the ability or has low ability to leach from the soil into the groundwater, thereby helping preserve Maine's good drinking water. It is not going to persist for a long time in the soil, so it's going to 90% of it will break down within 100 days, and it's not going to accumulate in the food chain. So in fish, 90% of the Tebufenozide is excreted after 15 days. 4 minutes? Yeah, I can do that. So in trout, less than 1% of the tebufenozide that's going to be and taken up by the trout, less than 1% will be found in edible tissue. Most of that would be found, so most of that remains is going to be found in the liver. So there's going to be hardly any risk to anglers. Just last night, I tried to provide some context to this. What does it mean by low risk to anglers who wish to consume trout? So I did some math, right? So suppose you catch a 2-pound trout, right? And so we're all thinking, well, the trout that we catch is much bigger than that, right? So suppose you catch a 2-pound trout, and that trout had ingested 300 milligrams of tebufenozide. First, that's going to greatly exceed the amount that's going to be applied, right? So it's a very, very conservative estimate. But I chose 300 milligrams because that's about the weight of an aspirin tablet, so we can kind of relate to it. So if that 2-pound trout consumes 300 milligrams of tebufenozide, the consumer would have to still eat 500,000 trout to approach a lethal amount. I mean, that's 500,000 trout in one setting. That's just not realistic, right? So that's one, so therefore, low risk to anglers. Lastly, additional information about tebufenozide. It's not carcinogenic. It's not a neurotoxin. In other words, it's not going to increase risk of neurodegenerative diseases such as ALS or Parkinson's. It's not an endocrine disruptor in humans. In other words, it's not going to disrupt hormones such as testosterone, estrogen, etc. And it received EPA's first Green Chemistry Award in 1998. I can speed through BTK quite quickly. It is a naturally containing spore, so it is not a chemical. It's a spore from Bacillus thuringiensis kurtaski. That's a mouthful, so let's just refer to it as BTK. It's an endotoxin that is dormant and is only going to be activated in the alkaline environment of a caterpillar. Let me rephrase. So the caterpillar's stomach is very alkaline, pH of 9. That's in stark contrast to humans and other mammals that have stomach pH of 2. And so due to these physiological differences, consumption of BTK is going to be not activated in humans, but will be in caterpillars. It's been used to control insects for 50 years, and it's been approved for organic crops in Maine. Less toxic than tebufenozide. However, consumption of BTK by non-target caterpillars will still be toxic. Half-life is 2 to 4 days. Overall, the risk assessment of BTK is low risk. Because of its big size, it's not going to leach from the soil to the groundwater, and there's no risk of bioaccumulation in the food chain. So lastly, in conclusion, both these active ingredients, very low risk to the environment. They're deemed practically non-toxic to humans, birds, most insects. It's anticipated that there could be some low risk to birds that prey upon caterpillars. This is especially relevant to the birds who consume caterpillars, right? So their diet, their foraging habits might change. And then lastly, the insecticides will be lethal to non-target caterpillars if they consume fir spruce needles that have been applied with these 2 active ingredients. Thank you.
Thank you, Doug. So far today, we've heard some of the background information and current status of spruce budworm in Maine, learned about the biology and monitoring efforts going on in the state, and strategies being used by our neighbors in New Brunswick, and a little bit about the chemical treatment options. Our next speakers are from the Maine Budworm Response Coalition. Alex Ingraham is the chair of the Maine Budworm Response Coalition, an alumnus of the University of Maine School of Forest Resources and president of Pingree Associates. And Ken Coffin has 30+ years of experience as a forester for Irving Woodlands. He is also the co-lead for the Maine Budworm Response Coalition tactical team. His family, friends, and coworkers in Aroostook County depend on a vibrant, healthy forest landscape. The jobs and recreational opportunities that it provides them are paramount. Over the last 5 years, he and his colleague, Chris Houston, have led 3 successful early intervention spruce budworm treatment programs in the same impact area we are discussing today.
So everyone's learned quite a bit about budworm today and early intervention, and so I will try not to eat up too much of your time, and hopefully the budworm do not eat up too much of the fir trees in our forest this spring. The other thing you learned is that Ket is much better at following directions in terms of creating a bio. Mine was short and sweet. Ket's was a lot more detailed and useful.
Regina did mine.
Oh, Regina, good job. So we're here today to, well, we're between you escaping here, so we'll be cognizant of that. But we're here to talk about what the landowner response is and the Maine Budworm Response Coalition and why we came together as a group. You've heard about all the risks associated with spruce budworm for our forests. And so we decided that given the abundance of information and research that has gone on in New Brunswick, given the spruce budworm outbreak that was predicted for 2025, that was an example to follow instead of continuing with a foliar protection study, foliar protection methodology that had been used in the past. So why did we think this? Because the repercussions of a spruce budworm outbreak from an economic standpoint, never mind a forest health standpoint, are massive. We're talking 494,000 cords per year, an economic impact of roughly 800 million a year, and impacting 3,865 jobs. This is on top of the recreation, the enjoyment of the woods and keeping a healthy forest. We also need to keep a healthy forest economy. So as you heard earlier today, there's a solution that was developed in New Brunswick in partnership with Quebec from lessons that they learned, and that is the early intervention strategy. As you heard from Angela, this is predicated on good data and early detection. We need to know where the populations are in order to identify those hotspots that Sara talked about and showed illustrations of, so that we know where the areas that need to be treated are. So that comes into the early intervention section where we target insecticide treatment to prevent spread and prevent this hotspot from growing. It is really important to think about this in relation to wildfire, not just because it's a risk that is created if we do nothing, but also as an example of how you treat this. You treat this like a wildfire, you stop the spread. And then lastly, communications. What we're here today to talk about is to make sure everyone is widely aware of what we're doing, how we're communicating, and talking to both our constituents, our stakeholders, and the general public as well. So Ken, I'll let you fill in the gaps that I missed.
Yeah, so great job. We've learned a lot today, just listening to everybody. Early detection, that's what it's all about, EIS, we're on it. On the early detection side, just like Alex said, I'd like to think of it as a forest fire myself. It's looking for smoke, and on the early intervention side, it's mapping it out, pinning it down, narrowing down the target. Where is it? What is it? Plan for the risks. That's a big part of what we've been involved with over the last 2 months, I'd say, is risk planning, habitats. What are they? Where are they? What do we need to protect? Water, we heard Doug just speak to that. Identifying the water sources, people, proximity to where we'll be doing the treatments. And then a big thing is just leveraging. There's a lot of knowledge in this room. I see people that have been involved with spruce budworm in the past and more recently, and then there's people like Sara, we just heard from New Brunswick to leverage. So taking all their knowledge, looking for the right products that Doug just spoke of, that'll help us minimize the risk, using tools and technology, things like autonomous spray systems in our aircraft to make sure that we're doing precision applications, applying standards, applying BMPs based on all of that knowledge, and of course, making sure that we're following the law Pilot safety is paramount, public safety, there's a lot of public that utilizes these private lands. We want them to continue to use it, but we wanna make sure they're safe. And worker safety is paramount, so a lot of us are undergoing training right now to make sure worker protection standards are followed and whatnot. I've had the privilege of leading 3 successful EIS treatments in Maine over the last 5 years. In the same areas that we're gonna be looking at treating today. On the communication side, broad audience, publications, Maine Professional Guides Association, I could list a whole bunch of folks that we've reached out to, businesses, users groups, and of course the agencies. They've had this thrown in their lap. We've all had this thrown in our lap, unfortunately. But everybody's worked together really hard to try to make things work on time here. Our primary focus though, in the areas that we're gonna be treating is North Maine woods, local ATV clubs, neighbors, Friends of Cross Lake, which has been a great platform for us in the Cross Lake area. And of course, individuals. We meet with individuals almost daily, people with questions, so we're really trying to take an active role in communicating with everybody that we can.
And so when we identified this problem late August of last year, the landowners and Maine Forest Service and our groups came together to devise what would eventually become the plan that we will show you in a little bit. I really want to thank the efforts of Senator Collins and her team. Have been instrumental in securing us federal funding for this project year. She secured $12 million for us to help pay for and treat this problem. And so now we're going to start looking to future years, and this is not unfortunately a 1 year one and done problem. This is something that we're going to have to continue to actively engage on. And so really want to thank Senator Collins, her staff, our entire delegation, and then the efforts of the Maine Forest Service, the University of Maine, and Governor Mills for continuing to put support behind this project and make sure that we can really keep a healthy forest and a healthy forest economy. So you've seen a little bit of this before. I'm just going to show a brief animation on where the budworm in Quebec has been, and then just watch New Brunswick, similar to Sara's presentation earlier. So this is from 2013 to current. And you can see that EIS in New Brunswick, it'll pop up and then it'll go away, pop up and then go away. That is the EIS treatment, the early intervention strategy in full effect. You can see the scary one, 2021 was a lot of red.
Real scary.
And so this is where we are today in 2025. This is the anticipated outbreak for this season, which is only a month away. So you will see now another graph. I'm sorry for the repetition. I won't repeat what's on the right. You've already seen that. But it is important to note on the left that New Brunswick, since 2014, has treated 1.6 million acres. In historical past treatments during the last outbreak, 1 to 3 million acres were sprayed annually. So over the last decade, they have treated basically what used to be treated every year. This is the big difference between early intervention and foliar protection, is it is highly targeted. So that gets us to what is the plan for 2025. Ken can talk about this a lot more specifically, but I'll cover the high level. On the left, we're dealing with the large outbreak that is coming over from Quebec, and then on the right, around Fort Kent is Cross Lake and Square Lake and the outbreak over there that Ed has been having fun with for the last couple of years.
Coming from Quebec.
Also coming.
Yeah, so like Alex said, it's 2 distinct areas. Sorry about the map here. It's this is our access control map. The scale, we can't zoom it in here for you, but it's 240,000 acres, Cross Lake area to the right, and on the left side to get up against the western border. That's another spot. We've had treatments in both of those areas over the last 5 years. The scary thing for me right now is if you get in a plane and you fly up against that western border. I think Mike showed a picture of it. You look across, we've got the green there, that's our treatment area, but if you look off to the west into Quebec, it's pretty scary to look at. So it's right up against us, and the prevailing winds come into Maine, we're up against it until that runs its course. We expect that our operations this year, depending on bugs and bud timing and the weather coming together, we expect that we'll open up our operations probably late May through June, hoping it doesn't take any more than 3 weeks, but it's very sensitive to the environmental conditions, heating degree days and whatnot, bug development. Access to the land is gonna be tightly controlled. So if you're gonna be in those areas, plan for road blockages. You can see some points on the map that indicate where some of those access control measures will be. If you're traveling in North Maine woods, it's a little easier. Check with North Maine Woods before you go. They've got a website and they've got professional staff. They're in Ashland and they can answer questions and keep you up to date if you've got plans to travel to North Maine Woods. The project reports. So one of the things we want to be able to do is report out on status. When we open the program, when we close the program, and probably some various updates in between. You know, we've got various sites where we can, where you can access that kind of information. Maine Forest website, Spruce Budworm Task Force website are probably the 2 places that I'd be going. And yeah, that covers a lot of area.
So in conclusion, we've looked at the research that's been done over in New Brunswick, the results from New Brunswick, and feel that this is a solution given the fact that Maine has gotten very lucky over the last decade to not get a huge influx of budworm, that we have the option of doing the early intervention strategy. So we are implementing that in 2025, and basically we want to make sure that we can protect the forests that are currently at risk. Use proven science that has been researched over in New Brunswick, and that we really want to make sure that we're acting at the appropriate time, and the time is now. So funding and commitment from all partners, both state, federal, and private, have come together to make sure that this project can go forward. We have engaged all the partnerships. You have heard from pretty much the entire engagement team today, from the Maine Forest Service, through the, Doug, I'm gonna butcher your group's name. Maine Board of Pesticide Control, thank you. Logan for the assist, I'm terrible with acronyms. And then we added another acronym, MBRC, Maine Budworm Response Coalition, to round out the acronym names. So really appreciate the time today that everyone's given us, and to finish it up, we'll put another picture of 1813. It's a popular town.
Yeah, it is.
Unfortunately, it's popular for budworm too.
Joe.
All right.
Thank you, Allison and Kay. At this point, I'm going to ask all panelists that are here to make their way to the front table. I'm going to be working screen tech right now and move the podium microphone over to Regina. We'll start with some of the questions that came through Zoom. We'll then open up the floor here in Orno to additional questions, and hopefully get to maybe a question or two up before Ken as well. We likely won't be able to get to all the questions today, but there is resources available where we will be keeping track of questions that come in and posting responses once we have good ones to provide. And that will be on the Spruce Budworm Task Force website.
Thanks everyone for that background information and giving us some context for what's potentially going to happen this spring and summer. So some of the questions we got on Zoom are related to the insecticides. So the first one is, how often will Tebufenozide be applied in the state as a preventative in a given area? Will it be rotated around the state? Are there years that no applications will occur? So I think asking about product use and how you're going to stagger BTK and Tebufenozide.
So from the perspective of the insect population, that's gonna determine where treatment areas are set each year. And it's not anticipated that the same exact acreage will be treated in any given year, or any consecutive years, I guess.
Yeah, that pretty much echoes what I was gonna say, and I would look to Sara, if she's still with us, to key in on what New Brunswick's experience has been. But from my recollection, it's just what Allison said, that it's not necessarily the same treatment year, and I believe Sara's presentation showed that as well.
Great, thank you. So knowing that spruce budworm outbreaks often last for years, how will we know if early intervention worked? How will we measure its success in Maine?
Yes, I'll take that. So continued monitoring. We're gonna go all of the sites that we did this year that showed those L2 counts. Some of them were very high in that region by Quebec. It will be treated this spring, and then we're gonna look at those populations again. There are also areas that aren't going to be treated, so that's kind of our control population to compare to our sprayed sites to see whether or not we have efficacy.
Awesome, thank you. So it looks like there are some questions about Tebufenozide and wildlife, specifically questions about risk to birds and spraying at the same time as fledgling season, knowing that birds eat caterpillars, feed them to their young. Just wondering if someone can speak to that.
I can do my best. Birds that prey upon caterpillars will be affected. Their diet will likely shift. So I can reference one study. One study looked at the effect of foraging habits or a lot of different parameters actually of these birds in a forest that was and was not sprayed. There was no effect on survivorship in birds. However, clutch size decreased, and I want to say the average weight of the offspring decreased. But again, just to restate that there was no effect on survivorship, both of the parents as well as the offspring.
Great, thank you. So there's some more questions specifically about communications and how folks will be updated as to when the spray is happening, especially in areas in the St. John Valley near populated places.
Yeah, so one of the requirements under the BPC rules are that we have to notify abutters. So over the last few weeks, hundreds of letters have gone out or been put together to go out to notify our abutters. And like I said before, the normal communication that I'd listed previously, businesses and whatnot.
And then we'll be updating the website from time to time, both at the start of the operations and then also during, and then when they're conclusion. Yep.
And I'll also just point out that when the Spruce Budworm Task Force was formed, one of the things that came out of that was website sprucebudwormmaine.org, which was set up to serve as a clearinghouse for information across organizations.
So for folks on Zoom, I think Erin has put those links in the chat. There's also handouts in the room to direct you towards those websites so you can find information as the season rolls out. So this is a question about aerial applications. How can aerial applicators ensure spraying won't contaminate adjacent properties, including water bodies and streams?
So buffering, it's a big one. Buffering all the risk items that I listed previously. A big part of it is hiring professional applicators that have got autonomous spray systems on their gear. We're talking, this isn't 1950s, 1960s technology, onboard GPS, 1 foot accuracy, searching for patterns within flight lanes, computer capabilities to make sure that the operators stay on track. And those operators, by doing that, have more time to think it's safer for them. They can think about other things because that autonomous spray system is very precise.
Yeah, like anything, it's about the data that goes in, and the more precision data and the better.
Okay, we'll try and get that fixed, but maybe just use your teacher voices in the meantime. Okay, so there are some questions on budget breakdown and the funding that's been awarded for suppression of spruce budworm. And so this person is asking, how is the money being itemized? There's $12+ million proposed. Some quick math puts the project at a maximum application rate around $1 million. Where is the rest of the money going? Are the planes and helicopters and labor already state-based?
Is this working? Okay, so I can talk about that a little bit. So sort of what we hear in cost, estimating costs for aerial application of pesticides. The ballpark figure for that is around $45 per acre for treatment, and that's actually what this project also came in on. Not through a contract that the state holds, but through a contract that the landowners hold. And so I don't know exactly how that other calculation was made, but it's about $45 an acre, and we hear that across the board talking with our counterparts in other states as well. And then the question about labor and whether it's a state program. State aircraft are not part of this program, and as far as the treatment itself are not part of the labor as well. So there are costs well beyond that $45 per acre that have already been incurred by landowners, and also resources going into preparing for this program this year.
Okay, so we'll do one more question on Zoom before opening it up. So this person is asking a little bit about some of the historical context that Mike gave and how we responded then versus how we'll respond now. So saying experts agree now on this approach, and it seemed like experts agreed then. How do we know that this won't have unintended consequences?
Yes, well, that is the classic question, and although we've kind of entered every, treatment period, not knowing everything involved in the situation, I think we really can look at this situation as building upon that track record. So, yes, we can never know all of the variables involved, but I think we have a much better idea of a lot more of those variables now, for the current outbreak situation. So, well, yes, we all have to acknowledge that, there are those unknowns and some things can happen that we can't account for. We've eliminated a lot of those negative effects from past experience, and as far as chemical use, as time goes on, that knowledge gets better and better, like Doug was saying, and from learning from New Brunswick's experience with the strategy, having that track record to compare our efforts to is tremendously helpful for our planning and our approach. So a lot of the work that has gone into figuring out what to do has already been done ahead of us, and so we do have a good knowledge base to build off of and carry forward.
All right, we're going to transition to questions from the room. And I'm going to do some mic moving around, so just bear with us for one moment.
All right, I have a question. It's kind of two questions, and the second is maybe two parts. But so I guess I work mostly with smaller landowners and some of the folks who are based up north. So I'm curious as to what resources are available to those folks. What can they access? And the second part of my question, so I work for MAVCA, so obviously we work with a lot of organic certified farms. In the 70s, a lot of those farms lost their certification due to drift. So we want to, where we can prevent that from happening again. So I'm just kind of wondering about those precautions, and if the drift does occur, who holds the liability?
I'll take the second one. I forgot the first one. The second one, Mofka, you know, one of the first things that we did was to look, because we know organic growers' concern is Mofka has a great site with map of their growers, and I think the closest grower they have is, I'm gonna say 5 miles as the crow flies across the lake. It's somewhere out on the Lamberto Road on the east side of Long Lake, Pelletier Island. And we put out the notices to all of our butters. And we have to follow the label when it comes to the aerial application, which means monitoring wind and drift. And I have a lot of confidence in the applicators that we have undertaking this program to make sure they abide by the law.
So you heard 5 miles for this year. In future years, if there happens to be something that is near, then it will be buffered and there'll be conversations. And particularly, if they're comfortable with the use of BTK, which is organic certified, then that can be used in those regions as well.
Thanks, I had some help in remembering the first question because it went from my mind. So as far as small landowners, for the 2025 treatment year, there is not a program available for small landowners just because of the depth of the details that are needed on the ground before you can implement a spray program from the air. So details like stand composition and actual property lines, not tax maps, and all of those things. Our hope is that in future years, there'll be funding and also a program available that small landowners can engage in. Oh, right. Liability part?
This year, we do not believe there is a risk.
But if it, in the case that it does happen, where does liability fall?
It falls within, I don't know, that's a better question for legal. So.
Go ahead, look into that and find out an answer.
I think we have the person that has the answer.
Well, thanks for having me here. My name is Alex Peacock. I'm the director of the Board of Pesticide Control, and the liability falls with the applicator and the applicator company to make sure that the product is not tripped off the target site.
Thanks, Alex.
Thanks for the assist, Alex.
I had a question based on Dr. Mech's answer to one of the prior questions, and I think you had said that some like kind of control areas are chosen, and I was just wondering how those are chosen.
So they're not really chosen as much as like the map that you saw where the spray is occurring. If you overlay that with where the model predicts populations to be high, there are some pockets that are left open. So not every landowner in that region is a part of the spray program. There were a couple of companies that have opted out. And so those would be locations that would have those control populations.
Okay, that makes sense.
Yeah, a question, Tebufenozide versus BT. Is there a cost difference between the chemicals? And what's the determining factor for what is going to be actually applied?
Thanks. So there is a cost factor, and the cost factor is BTK for the efficacy to meet, so the performance of the chemical to meet the threshold that we've determined under early intervention strategy. You have to apply it twice. The chemical itself for a single application is very similar.
Sorry, Mike, I should have caught you while you were over here. Good exercise. Good. So it seems like a lot of the pressure from other populations is in Quebec, and given that these outbreaks last around 10-ish years, I'm just curious where Quebec is on that curve that Angela showed of the different outbreak stages.
Yeah,
I guess it depends what part of Quebec, but I would put Quebec at the uh-oh phase. Because they have experienced mortality, and have been for a while. So
How long have they been in the uh-oh phase?
I think this first year was in 2004 when they started experiencing, I think 04.
yeah, 04-ish is the
That's very
estimate.
big.
Yeah, so it's kind of dependent. So the trees right across the border. Yeah, those are the ones. So, but that I would put at the uh-oh point, as well.
It's really hard to talk about Quebec, you know, as
Huge.
in comparison to Maine. So there are parts of the province where the outbreak has collapsed, and there are parts of the province where the outbreak is building, and there are parts of the province where the outbreak is at its peak. But it is building in the area to the west of Maine, where we do expect to see some of the significant inputs from atmospheric transport of the moths.
And then one final note on that, land ownership in Quebec is vastly different than in Maine. The percentage of public or crown land in Quebec is very, very high and the resources are limited. So they're spraying or treating roughly 2.2 million hectares in Quebec this year for foliar protection. But it is predominantly on the north,
It's not close enough.
and closer to Ontario. But the area directly west of Maine is an area that has a lot of private lands, so the intervention strategy might be different there.
Thank you for all the great questions so far. If we haven't gotten to your question yet, and you're on Zoom, please keep putting the questions in there. We will compile all of them and formulate responses that we'll post on the Spruce Budworm Task Force website. At this time, I'd like to turn it over to Fort Kent, Neil Thompson, and see if there's any questions coming from the county.
But tell me at once.
I have one question on the buffers for the water, the protected resources. Does that 100-foot buffer include forested wetlands as well?
Yes.
Thank you.
We have more questions on Zoom, if there aren't any questions in Fort Kent. Okay, if there aren't any more questions on Fort Kent, I think we'll take another couple questions from Zoom. So this next one is, what is known about the strategies landowners who are opting out of this program may be planning to use? I'm curious if there are any landowners in the room today that might be opting out of the program.
So the one landowner that I know of that opted out for this year is got minimal acre exposed, and is watching what the success of what we're doing this year and gonna take a response on what their activities are gonna be next year. So more along the lines of watch, listen, see progress, and then formulate a response for 2026.
I can speak to that, but PNC and, you know, basically we wanna, you know, increase our L2 sample and stuff, and like Alex said, it's kind of right on our doorstep there, but we don't have a lot of acres right there, so we're certainly fully involved in this whole thing and watching it closely, and we'll definitely be involved when we feel it's necessary.
Yeah, I'll add that they've been an active participant in all our meetings and structure and know the full plan and are actually actively helping, so.
Okay, the last question is about timing and phenology. So what is the forest composition and treatment areas? Red spruce versus balsam fir? Two-part question. What timing relative to spruce bud, to red spruce bud covering and balsam shoot development is intended for BTK application?
Yeah, so BTK, all of the areas that are within 1,000 feet of dwellings will be treated with BTK. So those are called SALOs under Maine Board of Pesticide Control. So BTK will be the application that we will use in those areas. Most of those areas we've treated before with BTK. The second part of that question, what was it? Oh, yeah, spruce versus fir, heavy fir. You know, residual fir from the last budworm outbreak, 80%.
I think maybe one more important detail to add, based on some of the maps you saw. So those treatment areas are already clipped, so to speak, to the appropriate forest type. So it's not like the complete forest will be blanketed with spray with spruce fir mixed in. Those are, you know, stands that meet a minimum criteria of spruce fir competition. So you will definitely have separation of hardwood stands from conifer dominated stands that are the intended target of the spray program.
Yeah, thank you, Mike. We should have noted that when we talked about the map. I think that is all the time we have for today. On behalf of Keeping Maine's Forests, I'd like to thank the Spruce Budworm Task Force, Cooperative Forestry Research Unit, all of our panelists and presenters, and everyone who joined us both in person in Orono, in person in Fort Kent, and on the Zoom webinar. If you are sticking around for either of the in-person after options, I will close the webinar now and then we will discuss details for that. Thank you so much and have a wonderful day.